Welcome to Sunrise Session with USU, and where we bring in some of our finest researchers to share with you some of our insights and exciting times. My name is Mark McClellan. I'm the Vice President of Research and Dean of the School of Graduate Studies at USU. President Cockett could not join us this morning, but she sends our highest regards. Sunrise is, of course, just one of the avenues that we showcase our faculty's researchers, and we try to bring the best and brightest to interact with our Utah citizens. Another way that we do this is through a week-long celebration of research on USU's campus, and that's Research Week that begins April 9th through the 13th. Some of our biggest events there, we have our USU Ignite program. We have 9 of our student researchers presenting their experiences. We have our faculty giving some of their experiences and showcases, showing some of their books that they're working on and programs that they're active with. It's an incredibly exciting time. We do that all inside our campus library, which to my thinking is exactly where research should be based. And we hope if any of you can make it up during that week, you can look on our website and see all of the activities that are happening there. But today we're going to take you into the world of chemistry. And we're going to be hearing from Dr. Lisa Berreau, who is currently the Executive Associate Dean for the USU College of Science. But to me, more importantly, core to her heart, she's a professor of chemistry in the Department of Chemistry and Biochemistry. Lisa is one of those brilliant individuals that knows how to deep dive into the science and then step out and help explain, take those of us who are a little bit more lay to the science and take us into it and share it with us. And she has an intuitiveness of that is magical and fun, and I know that because I've heard her speak already a number of times. She also has an energy about her that once you want to be on her team, you want to be a part of the exploration of the science that she represents. Her group is focused on using nature-inspired inorganic and organic chemistry. I just gotta share this because I spent most of my master's degree working on anthocyanin molecules, and she was talking about anthocyanin molecules. It was like, finally, a piece of chemistry I know. And so it was a great time. Please join me in welcoming Lisa Berreau to the podium. Please.
I'd like to start by thanking Regents for sponsoring this opportunity and also thanking ORGS for this opportunity to come to this incredible audience and talk about work we do at Utah State University in chemistry. And I know that's a subject that for many of us can be overwhelming. I don't want it to be that way today. I want to have a conversation. I want to talk about a molecule we all know about. We've known about it since we were children. And so let's talk about carbon monoxide. And the second part of that title, a potential therapeutic. I'm guessing most of you looked at that title and said, this is a big disconnect. Okay? So let's get started. This is where we all are right now. We all think of carbon monoxide as a gaseous molecule that's dangerous. We think about it when we think about our homes, our cars. And so carbon monoxide is produced through the incomplete combustion of fossil fuels. And so that can happen in our exhaust pipe of the cars, in cooking, in our fireplaces, our furnaces. And because incomplete combustion is something that does happen, we have to be aware of our carbon monoxide levels. And so all of us, I hope, have carbon monoxide sensors within our homes. And as I was having a conversation with Paul a few minutes ago, your carbon monoxide sensor is set to detect a certain level of the gas over time. And so low levels of carbon monoxide, something like 40 parts per million, will take a long time for your sensor to detect. We'll come back to that, but that's because that is not a harmful level for humans generally. If a higher level of CO arises in your home, that will set your alarm off much faster. Now, you can almost think of the human body as an alarm for CO as well. Because if we take a look at humans and exposure, a human can be exposed to 50 parts per million of carbon monoxide over an 8-hour workday. That's the OSHA limit. Higher amounts of CO, though, will start to cause human health effects, and so slight headaches and other types of issues, and exposure times, again here, correlate with the amount. So higher amounts mean shorter times of exposure for potentially lethal effects. Let's talk about what happens when we breathe in carbon monoxide, our basic biology. So when we breathe in carbon monoxide, it's coming in with the air we breathe, as is oxygen. In the lungs, those gases are brought to the bloodstream, and they diffuse through. And normal oxygenation means that the red blood cells will pick up oxygen. When carbon monoxide comes in, it's also picked up by our red blood cells. Now, the issue for humans and our detection, in part, of carbon monoxide is that when it does bind to our red blood cells, oxygen can no longer do so. And so CO poisoning is thought, at least in part, to be due to the saturation of hemoglobin. Now let's go to a bit more molecular level here to talk about the effects of CO. If we think about just a drop of blood is red. That's due to the presence of the red blood cells. Within the red blood cells are molecules, proteins, called hemoglobin, and this is a chemical structure of the center of hemoglobin. Now, for those of you who are not chemists, I'm showing some elements written out in their letter form. Others I'm just showing as lines. And so the way to interpret that is if there are just lines drawn, it implies a large number of carbon atoms. And the different number of lines between them means how tightly they're hooked together. So you're going to see some more structures that have both atomic labels and just lines. Okay, so in one drop of blood, we have about 350,000 cells that are going to be oxygenated or are going to take up carbon monoxide. Within each cell, there are 250 million molecules of hemoglobin. So we have many, many sites at which the gases are going to bind. Now, how does that happen? That molecule, if I go back, if I take this structure now and I turn it on its side, what we can see is that the iron that sits in the middle is going to be the site where either our oxygen binds or carbon monoxide binds. The issue when we are carbon monoxide poisoned, as I said, is it blocks oxygen transport. And the main reason it does that is that CO binds 300 times stronger than oxygen to that iron center. And so once it's on, it's very, very difficult to get off. Okay, that is the basics of carbon monoxide chemistry that we have known for decades. This is how carbon monoxide in the literature was written about for a long time. The part that wasn't written about is the fact that that same molecule, that same heme, is actually a source of carbon monoxide in the body. It serves as a molecule that, when it is broken apart, will release CO. Now, let me give you the one visual of the day. If you're going to remember anything from my talk, all of us go out and do athletic activities, or we do something in the house and we get a bruise. And that bruise over a series of 3 to 5 days changes color. It goes from red, purple to green to yellow and back to our normal tissue color. You are watching chemistry occurring in that bruise. And the chemistry that you're watching occur is a CO release reaction. Let me walk you through that. That heme molecule that actually binds CO, when the body is done with heme, it breaks it down. It uses an enzyme called a heme oxygenase. The oxygenase means it uses oxygen. So the body uses oxygen to start breaking bonds and kicking out CO. So the first step of a bruise healing process is a CO release reaction. Now, there's also other products released, something called biliverdin and iron. The iron gets quickly scavenged up by the body because it can cause issues if it's left free. And then this second step of bruise healing goes to bilirubin. What we want to point out overall is that the body has a mechanism to release CO at the site of a wound. Well, it turns out with that idea, researchers over about the last 20 years have been thinking about why that's the case. Why would humans release a low amount of CO? So people started studies in cells, studies in small animals, and what is found is if you keep levels of carbon monoxide low, remember we looked at those thresholds early on, if you keep it low, what you can observe is positive health effects in some various models. So models for cardiac graft rejection, hemorrhagic shock, hypertension, and those low levels over periods of time can serve as producing some interesting effects. I don't have time to summarize the literature here. There are a number of studies, up through phase 2 clinical trial studies, of CO gas being used to treat a variety of conditions. Much of this work has gone on at medical schools, for example, at Harvard Medical School. And CO has been found to possibly be useful for all of these different types of medical conditions. Now, I will just note the nature of CO is such that there's a good and a bad CO. You can almost think of it this way. If you deliver a small amount, you might be able to do it for good. If you want to do it for bad, like to attack a cancer, you could also deliver a higher amount. So both sides of that are under exploration. Now, this is where I want to take you on a deeper dive now into how you would do that. How do we think about delivering CO and using it possibly therapeutically? Well, the first thing you're going to have to do is be able to work in that low range. You've got to be able to control the amount that you're going to give. So you're going to have to stay in what we'd call an asymptomatic range. CO can be detected in your blood through the measurement of how much is on hemoglobin, or through exhaling. You can get a sense of how much CO is present. Treatment effects are going to have to be done in what's termed the asymptomatic range, which would be less than 10% in the blood. People are thinking about ways to do this. They've started with thinking simply about CO gas, and I'm highlighting here two companies, Mallinckrodt Pharmaceuticals and Proteris, both of whom have developed delivery devices that are going to be able to control the amount of CO gas mixed in with other gases for exposure to patients. I'll just note both of these companies have been rapidly evolving. Mallinckrodt Pharmaceuticals bought out somebody else in 2015 to get this device, and Proteris is a company that recently merged with another company in development of this device. And so it's a rapidly emerging field. Now let's talk about that inhalation for just a minute. Let's think about that. So if you're going to deliver gas and you're going to deliver it in a controlled amount, we'll think, okay, it's coming into the red blood cells. But here's the issue. With the gas, you have absolutely no control where it goes. None. Zippo. So that gas is going to be exposed to everywhere that the blood cells will take it. I want to introduce you now to a new term, a carbon monoxide releasing molecule, or a CORM. This approach is thinking of delivering CO by using molecular structures to do it. So instead of having a gas where it's just going to diffuse wherever it wants to, we're thinking about molecules that would deliver CO. And so, for example, if you bring in a CORM, maybe through the GI tract, you might be able to think about, okay, I could perhaps avoid healthy tissue and go to a targeted site for CO delivery. This has some interesting prospects, and we think it's an area that deserves a lot of additional investigation. We think so because there's a lot of advantages to using a CORM, a CO-releasing molecule. For example, the dose control is much better because you can determine how much you're giving easily. Tissue specificity might be controllable. You can determine how much CO you put in, and you would be moving away from having specific equipment that would have to control the gaseous amount delivered. Okay, so now it's here where I take you back into chemistry. So if you're going to deliver a CO with a molecule, you have to pick out the molecular truck, essentially, you're going to use to deliver it. The early days of the literature in this field were centered on using metals to bring the COs along. That's because metals like to bind CO. We already saw the iron center in hemoglobin like to bind CO very strongly. So the early days of this field was about taking something that had metals in. How many people have heard of ruthenium? Probably not too many. That's what that metal is Ru. It's ruthenium. It's a heavy metal. This is molybdenum. And so people thought about, hey, let's use metals with CO attached to deliver CO. These molecules have been used extensively in the trial portion of CO-releasing molecules. But there's issues when you use a metal-containing CO-releasing molecule. They add complexity because when you start to think about putting a molecule into a biological system or as a therapeutic, introducing a heavy metal ion with its own chemistry, its own issues. We also use heavy metal compounds for chemotherapy. And so there's issues that go along with heavy metals. So we may have toxic effects. Byproducts from these are a big issue. They're potentially reactive with other biomolecules. And so this field, as of this stage, is thinking about it this way. If you're going to use a metallocorm, a metal-containing one, you almost have to strip it up in a jacket because that jacket might be allowed, let the COs leave, but you have to control what the pieces are remaining. So the chemists are thinking about, how do I put this into a polymer? Or how do I keep it hung to a protein or something so that the metal doesn't become a problem itself? So with that intro, I'd like to take you where we're thinking about this. And our thoughts about this, as Dr. McClellan pointed out, come from inspiration from nature. Our chemistry is inspired by what we see that nature has already evolved. Now, it turns out there's a naturally occurring compound called quercetin. And I put this picture up here because if you walk into Whole Foods or another health food store, you can walk up to a shelf and you're going to see bottles of this. It's a natural supplement. People take it. And it's a molecule with interesting chemistry. We know that that molecule actually, under certain conditions, is a CO-releasing molecule. Now, that hadn't been explored for anything health-related until we started looking into this. It's also known that quercetin is a member of a family of compounds called flavonoids. Flavonoids you may have heard of. There are commercials on the TV for certain types of flavonoid supplements. They're well known for being antioxidants in chocolate and in green tea and black tea. And so it's a family of compounds with very interesting properties. And what we're trying to do as chemists is harness this CO-releasing capability and be able to use it in a way for CO delivery so that our molecules are distinct from those metal-containing ones that I've shown you previously. So how do we do that? Well, before I go there, I just want to share with you, flavonoids are also well known in industry already. Flavonoids derived from fruits and vegetables, naturally occurring flavonoids, are used in the food and beverage industry, as I talked about, in some pharmaceuticals, in the animal feed industry. And so they're well known already in industrial applications. And so Our molecule, we think, is a very good one to think about for CO release. And I want to show you why quick. Our molecule mimics what goes on biologically. If you think about it, this CO release reaction for quercetin is very much like how the body is actually doing CO release. Again, the body uses O2 and iron heme to break down. Here we're using a different type of organic, but again, oxygen and CO is released. So this naturally inspired flavor again is coming through in our chemistry. And so now how do we really do this stuff? You know, this is all fun, you know, but how do you really do this stuff? This is me, and this is two of my students, one of whom is Marina Popova, who's right here. This is Marina, the back of Marina's head. This is Stacy Anderson. When we're in our lab, we think about how to conduct experiments to test ideas about how we can manipulate molecules and get them to do what we want them to do. So inspired by the quercetin that I've shown you, Stacy and I talked about how to make a flavonoid that we could use as a CO-releasing molecule and maybe make it light-triggered, so we had a lot of control over CO release. And so Stacy came up with an idea. She changed the structure of quercetin a little. She changed it by putting an additional ring on, and we took off some other groups, but it's close. It's pretty close. And so she made that molecule, and when you make molecules, you might say, what does a molecule look like? You know, what does a molecule look like? Well, that's what it looks like. This is a powder. This is a chemical that we make in the lab that's just a yellow powder, okay? And that yellow powder, we then said, can we make it CO release? Will it do the quercetin chemistry? So we exposed it to O2, and we gave it some light. And sure enough, we got CO to come off, and the oxygen atoms, if I just go back, the oxygen atoms that come from O2 end up in our product, and we can track them. So just like in biology, we're using oxygen to then release CO, and in our case now, we've got triggered systems. They don't just let it go when they want, we have to tell them when to release the CO. The benefits of our types of molecules, we get a whole amount of CO. One comes out of there. We get a 96% yield. These are really controllable in that, as I've said, they won't go off until we've got them where we want them. And they're stable. We can put them in all kinds of solutions, in buffers, as I'm going to show you, in cellular environments. And they're perfectly fine as long as we keep the lights away from them at certain wavelengths because they are light triggered. So let's look at light and how we can see these molecules. Molecules that are yellow, we see yellow because of the absorption properties of the compound. There's also something called emission from compounds. Emission is when molecules absorb light but then give off a different light. And so we're going to look at now something called emission. And I'm going to walk you into a cellular experiment here. This is a group of cells that have been exposed to two things. One is a blue dye that will label the nucleus of the cells. The other is green. That's our compound fluorescing. So we can see when we put our molecules into cells, and we can follow them. Now, can we release the CO in the cells and see it happen in real time? So let's expose to light. So over 3 minutes, we see a bit of a change. We see a little bit of distinction in the green color, maybe a little bit of fading. But then let's leave the light on a little longer. And when we do that, the green color disappears. So in other words, our CO-releasing molecules are trackable. We can put them into a cellular environment and watch the chemistry occur by watching the loss of that color. Let me show you a different way to do that. This one's really dark, and I apologize for that. We can track the green emission, as I said, but we can also put in another sensor, which is red, and you're not seeing it very well here, but we can actually detect the CO by using a different molecule in combination. And so our molecules are giving us the opportunity now to really look at how we can use CO in cellular environments and really track it carefully. Our naturally inspired CO-releasing molecules are advantageous, we think, for a number of reasons. We hadn't discussed until now, but they're generally non-toxic, and the byproduct is especially non-toxic. I didn't want to put up a lot of structures for you and things, but the bottom line is our byproduct looks like aspirin. It looks like the structure of aspirin. There are also other properties in this class of compounds, like anti-diabetic and other properties that have led them to be used, as well as some anti-UV properties that have put them into cosmetics. And so the whole class of compounds we think is quite interesting and worthy of further pursuit. And so as chemists, then, we start to play. And we like to make things. And so one of the things we like to make is compounds that we think might add advantages to our further work. And so one of the advantages we wanted to look at was, can we shift the colors of lights we use to trigger our CO-releasing molecules? My student Stacy Anderson made this family of compounds where, by changing just a few of the atoms that are highlighted, she can change what wavelength of light we can use to trigger the CO release. And maybe I'll just stop there and teach you the second walkaway point today. The second walkaway point, has anybody ever put a red laser pointer up to their hand or a green one? Okay, red penetrates your skin better than green. So you can, with a red laser pointer, a strong enough one, you can see it come through on your hand because the tissue in your hand doesn't absorb the light. Blue light or green light would get absorbed. Our first class of CO-releasing molecules needed blue light to go off. If we want to use them in a situation where there's going to be tissue present, we need to shift the wavelength of light that we're going to use so that it's useful. So Stacy's idea was to make molecules that would absorb red light. And that's what she did here by just simply changing the structure ever so slightly. In doing that, she found that the molecules actually absorbed more light, which was something we hadn't immediately anticipated would happen, but they absorb more light, and when that happens, they go faster. And so we get control about speed of CO release. Now, I want to walk you through now thinking about if you're going to put a small molecule into a human and how that molecule will behave. A small molecule will often be picked up by something else, like a protein. And the protein I'm showing here is called human albumin. It is the most common protein in human serum. Albumin is a well-known transport protein for many, many small molecules. Within albumin are binding sites. Those binding sites, as I've highlighted here in color, will be the places where the molecule goes and sticks on. We've discovered that we can change how sticky our molecule is to albumin. Marina Popova from my lab has recently discovered, and you guys are getting unpublished work, so this is new. Marina Popova has recently discovered that when we simply change an atom from an oxygen to a nitrogen, we change the affinity for albumin by a huge amount, from 1,000 to 1,000,000. These molecules bind at the same site that ibuprofen binds to albumin for transport. And what's really cool about that is it means we might be able to use our molecules in combination with proteins as delivery devices. There's a lot of interest in the literature right now of using albumin proteins to deliver drug molecules to specific targets. Our molecules exhibit many properties which we think are really nice for development of CO-releasing molecules. As I've shown you, we can change their structures to change their properties. They're non-toxic. We can trigger them. We have great control over CO release. And we can tune how fast they go by changing their structures. How might we use molecules that release CO when you trigger them? Well, you're going to have to be able to deliver the triggering device, so light. And so we think of our CO-releasing molecules maybe starting in topical applications or localized treatments or used in combination with existing technology like fiber optics. And our molecules, therefore, will be very specific for site and local generation of CO. I will contrast that with my fellow researchers in the field who are working on more systemic type approaches, where a pill would be how you would deliver CO that would have a release rate of a certain rate. Our compounds, as I've shown you, which is fun working in naturally derived chemistry, our molecules are actually really easy to make. And so I incorporate a lot of undergraduates in my lab. I can bring in a sophomore who's had organic chemistry, and they can make this family of molecules because they take, my students are sitting here, 1 day and a couple steps. So they're very easy molecules to make. As we've talked about, they're highly controlled and non-toxic. How do we think about this in the big picture? Well, we've patented our molecules. We've patented them because we think this field has a lot of interesting chemistry to explore. We think they're some of the most novel compounds for targeted delivery of CO. And our next set of goals is to show that we can localize them both within the cell, which my student Tatiana has a beautiful set of work that we're getting ready to publish on how we've localized them to mitochondria within cells. And also moving on to looking at animal models where we might see how our localized CO-releasing molecules actually work in an animal system. And so with that, I would just like to note that my lab right now has the benefit of being funded by the National Institutes of Health. This project really got its legs, though, at first from the National Science Foundation in basic science. And USTAR also contributed to an aspect of this that actually went off in a different direction. I will tell you, there's a lot of CO used in industry. We use it in giant tanks to do chemistry, and so if there were better ways to introduce it in controlled amounts, it would be good for industry as well. And so we have some project ideas there as well. And with that, I would like to thank you for your attention, and I'll be happy to answer any questions.